Today we are going to talk a little about epistasis. Epistasis is the interaction between two genes where one gene depends on the other in order for it to be expressed. Epistasis results in two or more genotypes sharing the same phenotype. So let's say we have two genes, A and B. Gene A is responsible for hair pigmentation. A capital A allele leads to the creation of pigment or color in the hair. A lowercase a allele leads to no pigment being produced. Gene B is responsible for hair color. A capital B allele leads to red hair, and a lowercase b leads to pink hair. So we would expect a person recessive at both genes to have red hair, but because these genes are epistatic, their phenotype would be white or colorless hair due to the A pigmentation gene. Remember, in epistasis, two or more genotypes can share the same phenotype. So assuming this is an example of a type of epistasis known as single recessive epistasis, we can draw a diagram to demonstrate the pathway from genotype to phenotype. We start with a colorless precursor. If the person has two little a alleles, they will stay at this point and will have colorless hair, regardless of what is happening at gene B. However, if the person has at least one capital A allele, they are able to produce enzyme A, which leads to a pink intermediate. If the person makes it to this point and has two little b alleles, they will stay at this point and will have pink hair. But if the person has at least one capital B allele, they are able to produce enzyme B, which ultimately leads to the red product, and they will have red hair. So now that we know a little bit about epistasis, we can now do a dihybrid cross to determine the genotypes and phenotypes of the progeny. First, we will look at a cross between heterozygous parents at both genes. We need to do a dihybrid cross because we are looking at two different genes. Once we complete the cross, we can then look at each genotype and determine the phenotype based on the epistatic nature of these genes. We can then complete the chart based off of these four genotypes, and we can see that we have 9 progeny with red hair, 3 progeny with pink hair, and 4 progeny with white or colorless hair. The ratio of 9 to 3 to 4 is expected for this type of epistasis. Let's look at another cross between big A, big B, and little a, big B, little b. Same as the last example, we can complete the dihybrid cross to determine the genotype of the progeny. Then, following the rules of epistasis, we can determine the phenotype of each progeny. Then we can complete the chart and see that we only get 2 phenotypes. Half of the progeny will have red hair, and the other half will have white or colorless hair, even though they have different genotypes. Thank you so much for watching.